Aloha and welcome to our video review for lesson 6.2, the cell cycle and mitosis. In this video, we will identify the phases of the cell cycle. We'll talk about what it is as well. We'll discuss the activities of each of the phases of mitosis, and then finally we'll take a look at eukaryotic and prokaryotic cell reproduction and how it differs a little bit. Okay, so let's look at the cell cycle. The cell cycle, quite simply, is the stages a cell goes through from when it's first created to when it splits. Okay, so if it were to die somewhere in between, obviously it'll stop. But basically, we're looking at when it's made to when it stops. So we can kind of see a nice little picture here, and we'll go through the different pieces and parts of this one here. The first stage that we want to talk about is what we call interphase. And interphase is the longest phase of the cell cycle. It's where the cell is doing what normal cells do. So if it's a muscle cell, it's behaving like a muscle cell. If it's a stomach cell, it's doing what stomach cells do. So that's what interphase is. It's this intermediate part where it's kind of doing what it's normally doing. And we break interphase down into 3 different stages. So we'll talk about those ones here. And you can see this interphase is marked right here by this big long arrow here. So you can see how it's really the longest phase that a cell is in. The first stage of interphase is what we call G1 phase. And G1 is its first growth stage. And this is where the cell does normal functions. This is right after it's split, right after it's formed, but it has to grow in size. So it has to become a full-size cell, so to speak. So that's what it's kind of doing here. It's going to be doing what it needs to do, but it's also going to expand and get a little bit larger. The next stage is the S stage, and in the S stage is really critical when we talk about cell division. When we're forming 2 new cells, each cell has to have a complete set of DNA. So during the S stage is where we see this duplication of the DNA happening. So while it's still maintaining its normal functions, inside the nucleus what it's doing is it's copying all of its DNA and making another set. Okay, and this is really an important process for it to go through. The final stage of interphase is what we call the G2 phase. And during the G2 phase, it's the secondary growth phase. So it's already copied the DNA, it's become a full size. Here it's going to try and get a little bit larger if it can. But also during this part, it's copying all of the organelles. And remember, the organelles are what's needed for the cell to survive. So it's copying the mitochondria, the RNA, the Golgi bodies, all that fun stuff. And this way, when it splits, each of the daughter cells will have everything it needs to survive. So in the G2 phase is where it's making these final preparations for cell division. This takes us to cell division, and cell division we can kind of see as this tiny little green arrow right up here. The first stage is mitosis, and in mitosis is where we copy the nucleus. We make sure that each of the nuclei that's going to be formed has all the DNA and all the materials that it needs. When that's done, then it goes into a process we call cytokinesis. And cytokinesis is where the cell actually splits and forms the 2 new daughter cells. Okay, so now let's take a little bit of a closer look at each of the phases of mitosis. We just mentioned that that's where we're splitting the nuclei apart. So now let's look at that in a little more detail. Okay, so let's look real quickly again at mitosis and cytokinesis. Mitosis is where we're creating 2 identical nuclei. So the process, the steps of it, the way that it goes is it makes 2 identical nuclei, 1 for each of the daughter cells. There's 4 phases in mitosis. They're prophase, metaphase, anaphase, and telophase, and we'll go into those in a little more detail here in a couple seconds. And then cytokinesis is just that process of dividing the cytoplasm into 2 cells. So let's go back and revisit interphase. Interphase is that long process, and during that long process, the cell's growing initially. It copies its DNA so that it can make 2 little daughter cells where it can split. And then finally it has that secondary, that G2 phase, where it starts copying all the organelles so it can go. And this is when cells are doing what they normally do. So it's a normally functioning cell. It's just getting ready for cell division. The first phase of mitosis is what we call prophase. And in prophase, the DNA thickens up to form these chromosomes that you can kind of see here. The chromosomes are made of 2 sister chromatids, and they're attached by a centromere. So each of the chromatids is, or there's one original and then a copy. That makes the sister chromatid. We notice that we have the centrioles up here. Okay, and in between them form these spindle fibers, and that spindle is really important for moving the chromosomes apart. And you'll also notice that the nuclear membrane is starting to dissolve away. Okay? The next phase is what we call metaphase. And metaphase has this M, and everything in metaphase is about the middle. Okay, the metaphase plate you'll hear about. What we do is we line up all the sister chromatids, line up across the center of the cell. These spindle fibers are going to start to attach to the centromeres right there in the middle. Okay, and this is how we make sure that everything gets divided up equally. If we line it all up in the middle and then we kind of split it from there, then everybody gets an equal portion. And that's what anaphase does. Anaphase is where we're splitting it apart, so you can kind of see how the spindle fibers are going to pull apart the sister chromatids, and they're going to pull them to opposite sides, okay? So to different poles in the cell, to different sides, and that kind of separates it out for us. And you'll notice during anaphase we start to see a little bit of a bend and a crease here, and that's where that cell's just starting. So cytokinesis is kind of starting, but it doesn't finish up until the very end, okay? So anaphase, we're just separating it out. When we get to telophase, telophase has that magical T for 2, and you'll notice that we have 2 new nuclei forming. You'll see that the chromatids are separated apart. This nuclear envelope is starting to form. The spindle fibers have dissolved, and we kind of start seeing what's shaping up to be our 2 new cells. You'll see that these indentations are getting a little bit bigger and like that. At the end of telophase is where we finish up cytokinesis. And cytokinesis is, like we said, just where we're splitting apart the cytoplasm. And that was what forms our 2 new cells. Once cytokinesis ends, we have 2 daughter cells. These daughter cells are then put right into interphase. They start doing what cells need to do. They get into the G1 phase so they can swell up and get to be a regular sized cell. They can grow. Then they'll enter the S phase and copy their DNA, the G2 copy everything else, and then they themselves will start going through this cell division process. Okay, the last thing I wanted to show you is what happens in prokaryotic cells, so we'll take a look at that. Now, prokaryotic cells, if you can remember, they do not have a nucleus, okay? So they don't have those internal membranes and things like that. So their DNA is just free-floating in the cytoplasm. Because of that, they don't go through mitosis. What they do is they go through a process we call binary fission. Okay, in binary fission, the DNA is just going to duplicate and then separate to one side of each, or one set goes to where, or the other set goes to the other side, and then they split off into 2 daughter cells. So they don't do this fancy mitosis, making sure everything's all fine and separated out. They just kind of split. It's a little bit quicker, it's not quite as precise, and that's why bacteria can reproduce so quickly. Okay, so just as a final review, let's go through interphase and mitosis one more time. Interphase is where the cell's being a normal cell doing what it has to do. It does have 3 different stages of interphase. The first one is G1, and that's where you have the newly formed cells going to grow to be its normal size. Then it enters the S phase where it copies its DNA, getting ready for it to split into 2 daughter cells. The G2 is where it copies its organelles and it'll get a little bit larger, like almost doubling in size so that it can split. That takes us to cell division. The first part of cell division is mitosis, where we make the nuclei, so we're going to focus there. Prophase is where the envelope breaks down, the chromosomes are thickening up, we start forming the spindle fibers. Metaphase, we line everything up in the middle so we can divide it equally. Anaphase, we split the sister chromatids apart, and then in telophase we start forming the 2 nuclei. And at the end of telophase is when we have cytokinesis, and that'll split it into 2 daughter cells, and the whole process repeats itself. Okay, thanks for listening, and I hope this helps.